Okay, yeah, thanks a lot for having me. I mean, thanks really a lot to the organizers, Bernhard and Toshia, for putting this together in such a professional way. I mean, it's been, perfectly organized and such a great product to interact with. Yeah, so, it's great that I can follow up on Jinpei with my presentation today because the two are related in some ways. Switch. Okay, so, of course, not much introduction is needed with respect to the importance of social interactions, right? So, of course, there's mother-pup vocalizations, courtship, and territorial disputes. And also, of course, vocalizations are very important as biomarkers for a lot of, for model systems of psychiatric disorders. So, for example, SHANK3 mice, we've already heard a lot about, or FOXP2 deficiencies. So, but of course, there's some challenges in studying social vocalizations, and Jinpei has already alluded to this. They largely come from the fact that vocalizations are ultrasonic, so very conventional equipment makes it hard to record them very well. They usually occur, or a lot of them occur in close proximity between the animals. There's acoustic occlusion happening, and there's also echoes in the room. And, as Jinpei has already emphasized, researchers have devised various, ingenious ways of circumventing these, for example, by studying only interaction of sames where you then don't individuate, or, for example, devocalize some animals, or anesthetize them, or separate in some way, as we've seen yesterday in one of the presentations. But I think based also on the questions that we've heard yesterday, where often the question was, well, who was really vocalizing? There's definitely a need in the community to really pinpoint in a social interaction, where is one vocalization coming from? And, so previous approaches, maybe one of the first that I found is this seminal work by Neunübel et al. From 2015, which had an accuracy of about 38 millimeters. And then, in more recent times, both Jinpei's and our work followed up on this, coming to accuracy of about 14 millimeters, which, already focused a lot and, helped to resolve a lot of interactions. But here, as you see here, drawn roughly to scale, there's still certain interactions that are very hard to resolve at this scale. And also echoes, I think, are in particular a challenge to resolve with kind of conventional techniques of, cross-correlation-based techniques. So I think we agree that more precise techniques are really needed and useful, in particular to achieve a very high certainty for localizing even single vocalizations, and in this way provide a reliable basis for all follow-up analyses. Of course, we ourselves and others are sometimes, playing with the idea of maybe, philosophizing based on the shape of the vocalizations and then being able to know who's male or who's female in an action. And that partially works, as we've shown, but I don't think it's a very reliable way. And I think it's better to really focus on the acoustic properties themselves. So what are the main technical challenges for this? So I think, of course, we have, through AVSoft and other companies, we have very high quality microphones. They have fantastic quality, but, they're often big and, of course, costly. And so I think what I would be presenting is trying to overcome this by using a different type of microphone. So what I will be talking about now in the first part is to achieve millimeter precision localization using a technique called hybrid beamforming. To do this, we built this setup here, which is composed of, on the one hand, 4 classical AVSoft microphones down here. And then up here, we have a 64-channel microphone array, the so-called CAM64, which is produced by the company Sorama. And so this microphone array has 64 MEMS microphones, which are arranged in a particular pattern that is optimized for the localization task. And if you look here, then you already see why this maybe hasn't been considered so much previously. So here you see, a classical AVSoft microphone with a beautiful, response curve over frequencies. And then you see the slightly worse, curve of the Sorama microphone. So the question, of course, was, can we overcome this, or is this in any way useful for studying mice and interaction? And so what I will show you later is that, our standard setup where we have 2 to 3 mice interacting on a platform. In this case, we use it without walls because we thought this is the best way of avoiding echoes. But in our newer experiments, we also use foam walls, very similar to what Jinpei was showing. So here you see the spectrograms recorded from these microphones. You get pristine quality from the AVSoft microphones, and here you see essentially on the average spectrogram what you get from the CAM64. So when I saw this in the beginning, I was pretty disheartened because I thought, okay, we're losing a lot of the high frequencies. But as you will see, the average spectrogram does not tell the whole story, because if you look at the individual ones, then a lot of the cross-frequency, can be decorrelated, like the noise in individual microphones can be taken out by using correlations across them. So what we did in this first study is to then use a hybrid combination of this microphone information in order to kind of optimize the information that we can get from the two microphones. So here you see our pathway. So, we first rely on the AVSoft microphones here to detect USVs. And then we use our so-called SLIM method, which is sound localization of intersecting manifolds, to put a to intersect a number of estimates from all the microphones, and in this way get one single estimate, from these 4 microphones. Then we also use the information from the USVs to then compute a beamforming estimate. So that's an estimate of the sound field intensity, in the plane of the interaction platform, to see where the sound was coming from. And you see here, we get a pretty small localized dot of intensity, and then we do a second level of beamforming, on a much finer scale, where we then, get the sound field distribution. So you see, it's not a single point, but I think the main reason why it's not a single point is because, the snout is an extended emitter probably. So it's probably unrealistic to hope that we'll ever get a single point. But in order to improve our accuracy, probably similar to Jimpei, we then average over that point to get one single estimate from this. Then following up on Neunebel's work, we use this so-called mouse probability index, but a kind of hybrid version of this to combine the two estimates, and that then together gives us one location estimate, here mapped to the snout of one of the animals. So does it make sense? I mean, is it useful to use the two microphones together? So here you see on a log scale, what the estimates look like, and you see that, you see that, the CAM64 error here is around about 5 millimeters. The SLIM error is around about, I think it should be 14 millimeters roughly. And you see that, most of them are somewhere here, but then you have some which, for example, are not very well localized by the CAM64, but they are well localized by SLIM, and vice versa. So that means the hybrid strategy is working out in the sense that, you do gain something on the two ends. So why does that happen? Well, mostly because you do have, issues with high frequency vocalizations in the case of CAM64, but also sometimes the intensity is more clearly represented in the AVSoft microphones. Okay, so if we put this together, then let's first look at some ground truth data. So we have a speaker that we move around in this arena. And then you see here that the SLIM, approach in this way gives an accuracy of about 8.5 millimeters. And with the CAM64 and hybrid, we get something like 1.9 millimeters in accuracy for a speaker. So it's a very small speaker and has a very small membrane. So probably even a more precise sound source than the mouse snout. If you look at this in the case of mice, then you see here our accuracy for SLIM alone are on the order of 14 millimeters. With CAM64, alone we get to about 5.3 millimeters or 4.5, and in the hybrid mode we can then get a final accuracy of 4.8. So that the most important thing, I think, is this rate of vocalizations that we can actually use, which we refer to as selected. So selected means it passed a 95 or 0.95 significance criterion or certainty criterion with this mouse probability index. And I think this is one of the most important numbers, namely that in this interaction we're able to reliably assign about 90% of the vocalizations to one of the mice. If we then look at individual mice, so it's like a pseudo ground truth estimate. So we have some data from single mice and some data, from cases where the mice were far apart. Then we get something like, 3.7 or 3.4 millimeters with then, very, very high rates of assignment. And so just to visualize this a little bit, so essentially we get, a accuracy that maps pretty closely onto really the extent of the mouse snout here. What does that look like in practice? So here's a little video with downsampled vocalizations. And you see we kind of can closely follow the male mouse in this case, vocalizing a lot in the social interaction between the male and the female. So now, what this allows us to do is to come up with some spatial profiles of social interaction, maybe the most obvious thing you'd want to do in the beginning. So one thing that you see is, for example, if the male mouse is emitting vocalizations, what is the relative position of the female snout with respect to this? So you see there is quite a number of snout-snout interactions where vocalizations are being emitted, but of course there's also snout-abdominal interactions, and so that's why you have a pretty wide distribution of the female snout, relative, because they're in relative angles to each other. Yeah, I mean, if you, for example, we can also use the tail marker for the female mouse, and then you see a very strong concentration of male vocalizations being emitted at the female abdomen. Conversely, for the female mouse, we see a strong focus around the snout. But we will take this with a bit of a grain of salt, because at the snout interactions, of course, the most difficult to separate, because these are the ones where you're really in extreme tight conditions. Yeah, you also see this here that the relative number of female vocalizations is very small, so 323 out of the 7,000. So then, that raised for us the question, how much do female mice actually vocalize in these courtship interactions? And I do really want to emphasize, this is by no means a general statement about, female mouse vocalizations, because we've seen beautiful work where, under other conditions like female-female interactions, etc., there are a lot of female vocalizations, or for example, we also ourselves worked on data from Kurt Hammerschmidt with these anesthetized intruder paradigms. So evidently, female mice vocalize a lot. But one thing that we found is that in these courtship interactions is really very limited, and it also depends on the accuracy of your method, how many of them you actually see. So for example, if you look at, our less accurate method SLIM, then you see the female percentage here, shown in red, in comparison with our hybrid estimate is already substantially reduced. If you then look at the selected ones, so the ones that are safely assigned, then you see there's also a further reduction. And if we look at only the ones which are, for example, far, where many animals were far, it goes even further down. There's, of course, a confound in the far, close one, because if the female vocalizations are just happening in the near vicinity, then that could explain why they're happening here. But so we think this is maybe not pointing to a total silence of female vocalizations during these courtship interactions, but we think that has been overestimated a bit in previous studies, probably due to methodological limitations. So one thing that we then studied also was triadic interactions. So in triadic interactions, we either had 2 males and 1 female, or 2 females and 1 male. So overall, we I think found that just one result really stood out, namely that if you look at the dyadic, the dominant, so, we separated the male vocalizers, or also female vocalizers, in dominant and subordinate vocalizers. And you see that typically there's a pretty big gap between the dominant and the subordinate vocalizer. So it seems that for the male mice, one, is the talker, and the other one somehow subordinates to this. Yeah, there seems to be a sort of, where we don't say real dominance, but some immediate dominance established in this interaction. Yeah, so in summary, for the first part, so we've gone kind of from this previous estimate accuracy of 14 millimeters to something on the order of 3 to 5 millimeters, which we think, is a big step forward in trying to really localize and individuate vocalizations in this social interactions. And yeah, the 2 results that we have is that, this male and female mice have quite different rates of vocalization for sure, and also relative spatial profiles, as we could see. And then secondly, that one of the males dominates the conversation in this interaction. Okay, so at this point, you could say, fine, the system is running, and where do we go next? But I think there are some remaining challenges. So as you've seen, we have this integration of AVSoft and the microphone array. That is a bit tricky, to always keep in sync and precisely aligned, because if you want millimeter precision, of course, the whole system has to be really in millimeter alignment. And the second thing is that everything that I've shown you so far, has been post-analyzed. So we've done, visual animal tracking and acoustic tracking all post-hoc, probably more than a year or whatever after doing the recordings. And for most of us, it would be very convenient to have a system that operates in real time. So in many ways, the Live Mouse Tracker has done a lot of this work for you and us. But on the other hand, the one thing that is still missing there is the audio tracking. So what we then try to do is to come up with an extension of our system that allows this live audio-visual tracking, and using kind of a deep learning pipeline and beamforming in one combined system. So I will just quickly show you our architecture. So what you see here is a new device that Sorama is making, the so-called L642 device. And it's, it's very compact. I mean, similar to the CAM64, but it has, a nice visual camera inside here. And it also has a streaming API, meaning that, we can get data live from the device while it's working. And so in this case, we get video data at 720p and 30 hertz. And we also get audio data again at 64 kilohertz. And at the moment at 200 kilohertz, 64 channels at 200 kilohertz. We're hoping this will be expanded to 240 later. So then what do we do with the data? So one thing that's important for us is that we perform live visual tracking in this case. So we use the sleep tracker, which we think is a bit more performant than Deep Lab Cut. So this gives us live tracking while the experiment is running. And so this is really happening within a few milliseconds after getting the data. And then we use the snout locations that we have tracked in order to inform where we perform acoustic beamforming. So acoustic beamforming means that you can combine the information from the 64 microphones to get a virtual audio stream from various locations. And in this case, of course, we're interested in the locations which are the snouts of the animals. So, if we have 3 animals, we compute 3 different virtual microphone recordings from this. And then we use the audio segmenter in this case to detect vocalizations inside each of these audio streams. And then by comparing the relative intensity and which ones are contained in which of them, we then assign vocalizations per animal. And then lastly, we collect this information so that it can be directly made available after the experiment. So essentially what you would then get is visual tracking data and acoustic statistics alongside with this. So this is all running on a single GPU. And currently we're running at about 15 hertz. So that means you get 15 updates per second of this whole tracking routine. So first of all, let's again look at some ground truth data because it's a new device. So maybe, it has different characteristics. So overall, we again, perform our location estimates in different ground truth positions. And we end up with very similar estimates, of these, locations on the speaker. And now let's take a look at what the GUI actually looks like. So here we have, the basic GUI for initiating the recording. Then you have, the live video tracking here, where you see the tracking points, as you know them probably from Deep Lab Cut or other systems. Then have here this, this reddish glow here, which is indicating the sound field estimate superimposed on it. Then we have the average spectrogram, which is useful to look at, but as I said, right, it's taking away also some of the detailed information. And then we have the beamformed audio streams here. And so one thing that I really want to point out is if you look at this here, you still see the high frequency noise of the MEMS microphones. How you see it increasing with frequency here. But if you look at the beamformed audio streams, most of that noise is gone. Yeah, and that has to do with the individual noise contributions from the microphones averaging out in this targeted superposition of them. Then you also see here the USB detection showing inside here, and then you see how they align with the presence of the vocalizations. So one thing that you probably already see is, in this case, for example, it's animal 2, right? You see it also the tracker color here, where they are the loudest, and they're present a little bit in the other ones, but by relative intensity comparisons, we can then assign them to one of the animals. So here's an example of the recording and the tracking. So down here we have the vocalizations, and they're already color coded for which animal is vocalizing. So you see green here is one of the animals, yellow is one of the other animals. And similar to what Jinpei was showing, there is, in this case of pup vocalizations, there is a lot of intermingling between the vocalizations. But of course, these vocalizations are probably mostly directed towards the mother and not to the other pup in this case. Yeah, one thing to mention, I mean, so in this case, we did not, we separated the mother from the pups. I mean, of course, the goal later is so that we can study mother-pup interactions, and in that case, then, the mother would be freely moving, but the mother is so efficient in picking up the pups that essentially wouldn't get much data, if we combine them directly here. Okay, so then, just to a few cases, that Jinpei already mentioned are very important. Of course, if vocalizations are close, you can separate them. I mean, this is now close in time. So there is, no problem in terms of like close in time occurring vocalizations. And also similarly, if vocalizations are happening at the same time, then by using this beamforming in a frequency resolved way, you can also localize them. So here you see an example where one vocalization is here and then another one is directly above it. And by using only this range of frequencies and this time to localize, you see here that in one case it maps to here, and the other case it maps to here. Yeah, so that is really a great feature. And I think the method that Jinpei showed, this USB-sec, something that we haven't yet considered, but that is a great idea to combine these approaches. Yeah, so in conclusion, I think hybrid beamforming is really a nice technique that combines the strength of these two microphone arrays, achieves accuracy on just a few millimeter precision, and I think it opens really a new level of accuracy for studying social interactions. But as I mentioned, it requires this precise alignment between the microphones and the camera, so that is a challenge. And so that's why we think for the future it's going to be better to really rely on the single device system. And one thing that we really want to emphasize is that this local processing is important. So it's very performance, so you can get your data relatively close to the experiment, also including visual tracking. And overall, we see that the accuracy is quite comparable to the hybrid system. So we haven't evaluated this in direct comparison, but this denoising to higher frequencies really seems to allow us to also localize high frequency vocalizations. And yeah, as you've seen, so assignment of simultaneous vocalizations is possible, so that's quite promising. And this is also related to this denoising through beamforming. Yeah, so together that I think will then really pave the way to behavioral real-time tracking, and then also feedback paradigms where you can trigger, for example, a vocalization or another modification in your environment through a recorded and assigned vocalization. And so with this, I would like to thank the people who actually did the work. So Max Sterling was the one driving the hybrid beamforming project. This was in nice collaboration with Sorama and also Lucas Nolis from Nolis IT. And then the UltraX project, which is currently running, is mainly run by Ruben Teunissen, who's an AI master student, but has now continued on the project as a researcher. And Judith Homberg, who we collaborate with to apply the system to Shank3 knockout mice, and Freya, Olof's doctor, and Jasmine, who we work with to record these mother-pup interactions. And then Bram Stöller, who is also here, who is working for the company Brain Builders, who's now really interested in entering the market for these beamforming and acoustic camera technologies. Yeah, with this, I thank you for your attention, and if there's any questions, happy to answer them.
Thank you very much. Do you have any questions from the room? Well, I think the best way would be just to come here, or I will give you the mic.
Just a basic question about vocalization study. How you solve the problem in one case with interference of sound and dispersion effect. It is first question about sound. And the basic information was about the technology. How you make analog without conversion, and what kind of processing, because 64 microphones. You need simultaneously auto process power waveforms. Thank you.
Yeah, sure. So if I understand your first question correctly, you were asking, how we dealt with echoes or so in the environment.
Echoes is because the interference to sound, without putting out counting out one, get also interference of the sound and also dispersion.
Yeah.
The interference which makes the frequency, interpolation, and integration, and also other one false.
Yeah. So so the way that we beamformed was that we determined the extent of a vocalization in time and in frequency, and then we used this range of time frequency values to perform the beamforming. So it was essentially combining the information across the frequencies, and yeah, but that we did not see a specific issue. I mean, it definitely became worse if we, for example, included low frequency sounds, which may occur because of scratching or walking of the animals.
Would that mean two different numbers in one different sound?
Ah, that you mean. Yeah. Yeah. So I think, yeah, good question. So I mean, the interference. So we looked a lot for interference, whether two animals vocalized at the same time. But taking together the two things that we mentioned, that the mice, the female mice almost didn't vocalize at all, and the male mice, there seemed to be this dominance between them. I think that's the reason why we barely saw any superposition of vocalizations from two animals. Yeah, but that's why I think that problem was not a prominent problem. So we got this question also, from the reviewers during the review of the article, and so then Max actually went through the entire set of recordings and looked at every single vocalization again to see whether it's a good candidate for an overlap between two. But I mean, he found in the whole set of like 12,000 vocalizations, he found like 3 or 4 that were sensible candidates, but it was really a very minor occurrence. And then on your second question, so the technical processing, well, so lucky for us, that's all handled in the Sorama device. So essentially the digitization is all happening on the device. It's run, it runs a Linux system on board and has a DAC. And essentially that data is then transferred to us via a gigabit Ethernet connection. And so we receive the data digitized, and then we perform our analysis on that level. Yeah, I mean, so we stream the data to an M2 drive. So, I mean, in terms of like the data handling, it's really not a problem. I mean, it's about 60 megabytes per second, so that's quite doable. And in the in case of our second system, I mean, there we stream it to the GPU directly, and then the GPU processing is, all the pipelines that I mentioned are happening on the GPU, so that we don't need to shuffle the data around anymore. But we also save the raw data to disk so that it's available for afterwards.
Yeah, just wait. Okay, so the question was more than 3 mice in a home cage setting. In fact, this is also the question from the Zoom. Do you expect problems in a more real environment on the open platform?
Yeah, so I think, I mean, maybe so we have not tried more than 3 animals, but that's mostly, our capacity limitations. So we haven't, I mean, I don't expect any problems from having more animals, I would say. But regarding the more realistic environment, so I think one real strength of the sound field estimation is that you can get a complete picture of where the sound is coming from, and so you can effectively then eliminate echoes, by either ignoring these parts, or in our case by only considering the relevant positions, which are the snouts, and then make a direct comparison on this. So we think that really combats, the spurious echoes. But the other reason why we do it is because it strongly reduces the computational effort. Yeah, so essentially it's about a factor of 100 or so in terms of beamforming points that we need, and that's what allows us to then run at rates of 15 hertz or so. Yeah.
All right, thank you. So maybe just last question from Zoom. Just estimate how much costs the latest device. Just up to zeros.
That's a very good question. No, I mean, I think that the company Sorama is currently marketing the device at a cost of about, I mean, the device cost itself is about 5,000, but then they have a kind of a model where they charge you money for yearly usage, and so I think for 3 or 5 year usage is also about 5,000. Yeah, so for a 5-year project, you're looking at about 10,000 euros, which we think is still manageable. I mean, I guess if I mean, Avisoft microphones are fantastic, but if you want to build an Avisoft array of 64 channels, you would probably look at 100,000. So essentially, there is quite a drastic reduction in cost in the system.